Perfect. Security alert. Maybe is it like split screen? Oh, there we go. Perfect. Yeah. Does it want to do that? No, I can just stand at the computer too. Yeah, no worries. Yeah, some of these logos are weird, but okay, awesome. Thanks. Yeah. So thank you for the introduction. I'm Kylie, and I'm a student at Montana Tech this year. I'm graduating in 2 weeks, actually, May 4th. Pretty exciting. Thanks. Yeah. So my story with Phages starts back in high school. I was one of Steve's students, and you say that you're not responsible for me like excelling in science, but you made science super, super fun. And I'm very thankful for that because I wouldn't be here without you today. So yeah, I participated in BRIC in the phage digging program in high school, and then I applied to be an intern at Montana Tech as a Phages pipeline student and got accepted there. Made my decision to go to Tech pretty easy. After I got that internship, I also got a pretty sweet scholarship offer, so I'm like, yeah, I can't turn that down. And I continued after that summer during the academic year and worked on some fish pathogen, doing research on that. And then the following summer, I was a supervisor and had 6 awesome pipeline students. And yeah, they all did really well. I got asked in an interview recently, like if I had any struggles being a supervisor. And I'm like, honestly, no. Like they were awesome students and ready to show up and work and super interested in phages and learning more. And in the following academic year, I annotated a genome, Rialto phage, which I'll talk about towards the end. And in summer 2022, I spent my summer doing research at Montana State University under Dr. Diane Bimshock and working with organoids, and it was super, super interesting, really cool to be able to continue with the IMBRI program. And then fall 2022 to present, I've just been continuing on my FP research and yeah, been working through school. So the big title of my project, Isolation of Flavobacterium psychrophilium toward the development of prevention and treatments for cold water disease of native trout. So in simpler words, we want to generate a phage-based treatment for cold water disease of trout at the Washoe Park Trout Hatchery. So what is Flavobacterium psychrophilium besides a mouthful? It is a bacteria that causes infections in fish, and the disease presents as cold water disease. It's responsible for substantial mortality of fish in farm trout hatcheries around the world, and it exists in the water and in the fish, but it presents as disease when the fish are under stress. And strains of FP can kill up to 50% of the stock at the hatchery in any given year. And I'm going to refer to it as FP from now on, so I don't have to say the whole thing. Yeah, and it damages their organs. This is a pretty gross picture, I apologize in advance. But it eats away at their skin, damages their organs. It's just a pretty nasty bug, and we want to try to find a phage that can kill it. So y'all know what a bacteriophage is by now, I hope, especially my mom, because she asked me last week again. It's just pretty funny. So why would this be a good option to kill FP? Current treatments that they use are not very effective. They treat the water with hydrogen peroxide, and peroxide's not great on any living thing, as you know. And in high concentrations, it's pretty damaging to the fish and not super effective at killing FP anyways. Vaccines are not yet commercially available, and antibiotic treatments are not effective against FP prevention, because it doesn't always cause disease right away, and when it does, it kills the fish pretty quickly. So not great at prevention. And bacteriophages can be used to treat antibiotic-resistant infections. So where it all started at the Washoe Park Trout Hatchery. So this hatchery is in Anaconda, and it maintains a brood stock of West Slope cutthroat trout, which is Montana state fish, and it's native to Montana. It's actually the first state-run hatchery in Montana, and it was built in 1907. And they've raised tons of different species of salmonids. Arctic grayling, West Slope cutthroat trout is their main one right now. They do have a smaller tank for Arctic grayling that they're working on. Rainbow trout, brook trout, golden trout, lake trout, and brown trout. And they produce a whole bunch of eggs, and it is very financially damaging to have 50% of their stock be compromised by this bacterium, and not really a great way to kill it. So first, we have to get the bug out of the water and out of the fish. So we started by collecting fish and water samples from the hatchery, and we plated them on 2 different types of agar suggested by papers that we read. And we incubated them at different temperatures for a different number of days. It was a lot of just trial and error to get this bug out of the fish and onto a plate. And then we tested colonies using nested PCR. So what is nested PCR? Does everyone know what regular PCR is? To amplify a little chunk of DNA. So the first chunk that we amplified was the 16S ribosomal RNA, and we used primers 20F and 1500R and just made a ton of copies of that. And we used the product of that PCR into the second round of PCR using new primers to amplify a genetic sequence that is unique to Flavobacterium psychrophilium. So that is just a pretty quick and dirty way to just confirm that you have FP on your plate. And we tested plates that looked like this, which this is a pretty ugly plate if you know anything about microbiology. This was just a fish homogenate. We just blended her up and then streaked her on a plate. And obviously there's a lot more bacteria than just FP on this plate. And you can see all these little circles that we tested. Those were all these different spots. And then you can see the gel on the right where only 2 of them were positive for FP. So this stuff was hard to get out of the other contaminating bacteria. But we were looking for a 1089 base pair band on our gel, and we've got some positive controls down here. But we did get some positives, so we took one of those and we restreaked it onto a new plate. And we got a little bit of more isolated colony formation down here on the left. But as you can see, we tested about 10 spots on that plate too, and only one of them was positive for FP. So it was a bugger, it was a stinker. But we eventually got it isolated, and to confirm our PCR, I'm trying to think of the right word, the PCR, nested PCR and how that works. We got a plate from Ken Stegmiller at the Fish, Wildlife, and Parks, and we just have this one little colony on there and tested it, and it was FP, which is pretty awesome. So we were able to actually get it isolated from the fish samples that we took from the hatchery, but we had to do a serial dilution method to try and dilute all the other bacteria out of the way and get our FP to pop up on a gel. So now we have the FP isolated, what's next? So we want to optimize bacteriophage screening culture conditions. So any of you that have worked with SMEG, you know that you have to grow the bacteria in a lawn so that you can see the plaques on the bacteria to know that a phage is infecting it. And FP doesn't like to do that at all. Every time we tried to grow it in a lawn, it just like popped up with more colonies. We tried a bunch of different things, different concentrations of agar, and it was frustrating. And honestly, I was tired of banging my head against the wall. So we transitioned to bioinformatic analysis of the bacterium. We just want to learn more about it, see if it has any of these phage-resistant genes we've been reading about, and just compare it to other known strains of FP to see if ours is unique compared to ones that have been published in GenBank. So we started with analysis of that 16S rRNA gene, so it's the first part of our nested PCR, and we were expecting a 1,500 base pair band on the gel, and we got a whole bunch. That was pretty good. We selected 3 of these products and sent them for sequencing, and then we compared them with known strains using BLAST in GenBank. And we also made primers for a gliding motility gene that has known mutations to be phage resistant, and made some new primers for that and amplified that gene in our bacteria, but analysis is still in the works for that one. So for our 16S ribosomal RNA gene, we found that all of the strains that we had isolated, or that we thought were separate strains, were actually 100% identical. But I compared them to other strains of Psychrophilium on GenBank, and they're not identical to the other published strains, which is pretty cool. So we've got a unique strain of FP that is infecting the fish at Washoe. Yeah. And then this is what I've been working on this past semester. We found this bacterium on GenBank that has like 5 integrases in it, which is a lot, but an integrase is where a phage can insert its genome into the bacterial chromosome to be a prophage in there, and which we found the bacteria online had 6H, a phage, existing as a prophage in its genome. So we wanted to test if our bacteria had that prophage in its genome as well. And turns out it doesn't. The first lane is clear, there's no band formation in there, so we didn't find that same integrase in our bacterium. But it does have all the other integrases that were in the published strain. And those ones aren't necessarily associated with prophages, but I just thought it was super interesting to have all those same integrases as another published strain of FP from a totally different place. And yeah, some of our conclusions: a local strain of FP was isolated from Washoe Park Trout Hatchery, and serial dilution of the tissue samples prior to plating allowed isolation of single colonies. And nested PCR assays confirmed the isolated colonies as FP. And the 16S gene sequences of these strains were found to be 100% identical, but showed varying degrees of differences from the other published species in GenBank. And a local strain, or the local strain of FP isolated from Washoe, does not include the 6H integrase in its genome. So future work, we're gonna try to get this puppy to grow on a lawn. It's very difficult. And collect and test additional environmental samples to try and find a phage that kills FP. And eventually in the future, maybe have isolated tanks and develop a phage therapy like treatment plan for these fish to see if that would work. And then just continue analysis and comparison of the gliding motility gene product sequences with known strains using BLAST. This is Rialto, cool little picture of that phage. This is a SMIG phage. It had a genome about 57,000 base pairs. Yeah, I thought bioinformatic analysis was really, really cool. It was something different than what I had experienced in my prior phages experience. And all of it sitting at a computer, not as much in the lab, so a very different experience, but I learned a lot and I thought it was super interesting. And I've got some acknowledgements. Thank our funding, of course, and Ken for sending us some plates and helping answer questions when we needed him to. And Carrie Berger is the fish culture specialist at Washoe, and she was just fantastic through this whole thing. Gave us plenty of tours, let us take samples whenever we wanted. It was really nice of her. And then also my awesome mentors throughout the years. Hannah was my first mentor the summer after I graduated high school with Marisa and Chris. And yeah, all of them just have played such a pivotal role in my success and my scientific career. And I wouldn't be here without you guys. And some citations. And do you guys have any questions?
growing wands,
Mm-hmm.
and I'm thinking about how we were able to test the growth of bacteria using when I did mine, like the growth rates.
Oh, in a liquid culture?
So
Yeah.
is there enough phage action from a sample that you could see a difference in a rock?
I think we had trouble with that as well because FP doesn't really like to grow in liquid either, was the issue. It's just it's obstinate little guy. I don't know.
Yeah, it has a preference to be on a solid surface, but if we can get the culture to make thick cultures, which is probably the challenge to get the wand, is you need enough density of cells.
Mm-hmm.
Then you could do your experiment, Megan, where you look for the spectrophotometer to see a drop in OD, meaning the bacteria have lysed. But first you would have, it would, again, that is a great approach to have alternative approaches to a top auger long tail. And I should just mention that Carrie Berger, she came up to Raylin at a science fair. She had graduated from Montana Tech and she knew about phages. And she was having this challenge in their hatchery where she works. And she said, couldn't we use phages? Last week she got accepted to an interdisciplinary master's program to come back to Tech, work at the hatchery and isolate more bacteria and establish these conditions. So it's coming full circle. So you can hand it back to
Yep,
Carrie.
passing the baton backwards. Yep.
Again, the frustration of working through the crustaceans and then learning, sometimes you pivot.
Mm-hmm.
And that's not just true in science. But success comes from determination, bullheadedness, bashing your against the wall, and pivoting when necessary.
Mm-hmm.
So you
Yeah.
get fabulous project careers, just whatever you want it to be. Very interdisciplinary type of techniques there, and we're just excited that you stayed with us this long.
Yeah.
And,
I've liked hanging out with you guys. It's been pretty fun. Yeah.
Kylie, I just want to commend you as well because your resilience is amazing.
Thanks.
And I remember some of those days of we can't get it to grow, we don't
Yeah.
know what we're doing,
Mm-hmm.
and just persisting and pivoting is impressive, and it will serve you well in your career. And
Thank
your
you.
life.
Thank you. Yes, Hannah?
As an early phage hunter, probably one of the earliest in the room, as a little kid, did you ever imagine you'd be doing the type of research that you're doing today?
Definitely no. One of the biggest motivators for me to go into science, sorry, I might get a little choked up, but well, both of my grandmas passed away when I was really young, and one of them had cancer and the other one had early onset Alzheimer's, and I just wanted to find a solution for that, and man, science can get me there, and yeah, that was one of my biggest motivators, but I'm so glad that I chose this path, and I feel like I really belong here. And I was talking to some of my friends the other day, and they're like, wow, you've never changed degrees. Like the whole 4 years you've been here, you've just kind of, you stuck with it. And yeah, I knew this is what I wanted to do. And yeah, I'm happy I'm here. Yeah. Oh, sorry. Excuse me. Yeah, any other questions? Okay, sweet, thank you guys.